Now the leaves helping in vegetative propagation. So first we saw by stem, now we are talking about by or with the help of leaves. 2-3 very important examples which we normally talk of, or rather which we have been discussing from junior classes. One such example is of bryophyllum. Bryophyllum leaves are thick and their margins have such depressions, and at these points there is meristematic tissue. So here we many a times see a tiny plant with roots, so couple of leaves and small roots at these margins. These young plants or plantlets keep getting nourishment from that main leaf till their leaves are fully grown, and once this full mature leaf falls, all these plants start to grow independently. Other examples: begonia, centpolia, Kalanchoe. These are some more plants in which leaves help in vegetative propagation. There is a fern that is also an example of this. It is called Adiantum caudatum. It is a fern, and this fern is known as walking fern. It is called walking fern. The reason why it is known as walking fern is, in case of ferns, the stem is underground in the form of rhizome. What we see are the leaves. These leaves in case of this adiantum, the leaf bends and the tip of the leaf, when it touches the ground, it gives rise to the next plant. So from here arises a new plant and the leaves. Again the leaf would bend and the tip of the leaf, when it touches the ground, it gives rise to another plant. So it appears as if it is taking steps one after the other, and that is why this common name of walking fern. So bryophyllum is an old example that we always talk of. These are begonia, centpolia, Kalanchoe and adiantum, which are few more examples where leaves help in vegetative propagation. Now the third structure which remains is root. So vegetative propagation by roots. Roots are underground, they are for absorption, they can be modified for storage, but in some plants, along with all these functions, the roots also help in vegetative propagation. One example is of sweet potato. In sweet potato, on the roots, so the shape of the root is irregular. From the roots arise many other roots. So many a times there is a cluster of roots which we find growing. Same thing is seen in case of Dahlia. Dahlia is a flowering plant where again reproduction takes place with the help of root. So at the base of the root there are adventitious buds. So here there are some buds. If you talk about just how it is formed, suppose this is the first root. Here an adventitious bud is given out, and this bud again grows into a root. Again this root will have bud at its base and will give rise to new roots. And from every root would arise a different plant. So if there is one more here, so there would be 3. And then you can separate these, it would have its own root and its own stem. But the reproductive structure is the root, because the buds, they have appeared on the roots. These buds are called adventitious buds. So these are some examples where we find roots helping in vegetative propagation. Now this is all natural. Let us take some artificial methods. We, the students of biology, understood how the plant reproduces. What is required for new root formation? Node has to be there. Can we connect one part to another? So using that knowledge of plant anatomy, morphology, how it works, we have devised methods where we are using the same 3 things, either stem, leaf or root, to grow more plants. So those are called artificial methods of vegetative propagation. So let us take artificial methods now. Artificial methods of vegetative propagation. The first and the most simple one is cutting. It's a very simple method. You just cut this branch or stem piece of one plant, put it in the soil, and that will give rise to a new plant. But because we are doing it on a professional level or maybe in a scientific manner, we would see from where to cut. Suppose this is the stem and here are the nodes, and we want to grow this into a new plant. This is known as a stem cutting. You have just brought a piece or a part of a branch. Now we want to grow it. One thing which we have to keep in mind is that this node part where the meristematic tissue is, it should not get damaged. So we should cut it from a place which is slightly below the node. And instead of cutting it horizontally, the cut is always given in a slant. So the stem should be cut in such a manner that the nodes are not damaged and the cut is given in slant. The purpose of cutting it in a slant is that the tissue exposed is more as compared to the horizontal cut. Secondly, we dip this in some phytohormones. So after giving it a particular cut, then the tip should be dipped in phytohormones. Phytohormones are the hormones produced by plants which promote root formation, cell division. So, and the most preferred phytohormone is auxins. So these auxins are used. You just dip that slant cut stem twig into this phytohormone and then plant it wherever we want. Because of the phytohormone, because of more tissue exposed, because of the node intact, very soon from this node roots will arise and this plant will survive. So chances of survival, if you just put it in the ground as it is and if you do it this way, the chances increase many folds. So though cutting is a very simple method, but we are doing it in a scientific manner. The second method, again we are doing it, so it comes under artificial propagation. The second method is grafting. Grafting is normally used to combine the characteristic features of two plants or maybe more than two plants. In grafting, one plant gives its root system and the second plant gives its shoot system. Normally when we combine or we want to combine the two characteristics, we want a plant to have disease resistance, which is very common in wild varieties. Suppose we want to have a rose plant by grafting. We can pick the root system of a wild variety of roses. I am sure you have seen the wild rose plants. Those flowers are not beautiful looking. They are ugly looking plants. The petals are not very bright, not very attractive, but they have good odor or aroma. Plus, the root system is very strong. Normally, the wild plants are disease resistant because they live in the harsh conditions. Whereas, the rose buds which we purchase, they are beautiful looking, but they do not have good smell. Plus, they are very delicate plants. Every now and then you have to take care of them from various kinds of pests. So, if we can combine the disease resistance of one plant and looks of other plant, then we would get a plant which would have disease resistant characteristics as well as beautiful looking rose buds also. So, one plant would give the root system, preferably a wild variety. The other plant will give shoot system, which has the beauty of the plant or that ornamental value. So, now the root system, the lower part, it is giving us the root system. This is known as stalk. And the shoot system is known as the scion. C-I-O-N, scion. Now, how do we graft? There are multiple ways. One is known as, so these are the methods of grafting. We can take the stem and the root or the shoot and the root system of the same diameter. Suppose this is the scion, the upper part, the stem part, and then connect it to the root part. We have given slanting cut to both, and then this part is covered with soil, moss, and then wrapped around with a polythene bag, and here it is tied so that the tissue can fuse. And this plant, when it grows, it will have the characteristics of the root giving plant as well as the shoot. So, now we have combined the characteristics of two. This is scion and this is stalk. This is called simple angular grafting. One more method is when we give a wedge shaped cut. Here the diameters are slightly different. The root system is slightly wider and we give a wedge shaped or a V shaped cut, and in this we place our scion by again giving it a pointy cut. So, it fits into the root system. This is called wedge grafting. Here again you are going to cover it with soil and tie it with a polythene bag so that soil, moss, they do not fall off, plus the holding is also maintained. One more method is there in which if the base of the stalk is very wide and you want to have multiple grafts. So, if you take this as the root system, very wide, diameter differences are more, and here we make multiple wedge shaped cuts and we graft 2, 3, 4 different branches into it or the scions into it. So, this is one scion, this is another one, third one. This is known as crown grafting. So, let us name this. This is slant or tongue grafting. This is called wedge grafting, and this one is known as crown grafting. grafting is normally done to combine characteristics, plus grafting can also be done to improve the ornamental value. Imagine there is a rose plant with 5 branches and each branch produces a different colored flower. One branch is white, the other one has red, the third one has orange and so on. So that ornamental value of that particular plant is enhanced many folds. So importance of this is you want to combine characters of 2 or you just want to improve the ornamental value of that particular plant. For both, this grafting is very, very important and it can be very easily done. Rose is one plant where grafting is very successfully done, but we can do it with any other plant also. Now the next method of vegetative grafting is known as layering. Let us take layering now. The next method of layering, third, is also done in 2 ways. One is called ground or mound layering. Ground or mound layering, and the next one is known as air layering. This technique of air layering is also known as Guti. It is called Guti. Now let us see what exactly we are talking of when we talk of ground layering. This is done in plants which have flexible stem. So you can bend that stem and bring it down to the ground level. Normally plants like jasmine. In jasmine, the plants have very flexible stems. They are basically creeping plants. They climb, you normally tie them to some rod like structures. So now what happens in case of this jasmine, these stems, they are not very strong, they are not able to stand upright. So if you bring the stem down and cover a part of node with soil. So you select a branch, a healthy branch, bend it, bring it down to the ground level, and wherever you find healthy nodes, just cover it with soil. From this node region, adventitious roots will be given out, plus there would be a new plant. On its own, it won't bend and bury its node, but because we are doing it, it is an artificial method. What we have understood is that new node, roots or adventitious roots can be given from the nodes. Second thing, it is a flexible stem, so we can bend it. So here, because we are covering it with little soil or ground, we call it ground layering or mound layering. This is possible only if the branch is flexible. Suppose I want to do the same thing for a plant of say guava or pomegranate. These plants, or even citrus plants, these plants have woody or barky stems. So the moment you try to pull them down, they will come down to a little height and then they will break off. So it is not possible to bring that woody branch and bury its node or cover its node. So we have to do everything in the air itself. So we select a plant, it has its woody trunk and branches. We select a healthy branch, and on this healthy branch, suppose this is the branch which we have selected, we are going to remove the outer dead material. So after removal of the dead material, the branch is going to look like this. This was the branch, and this is the area from where the bark or the hard dead part has been removed. It is also known as ringing. We cut that portion and remove that ring of dead tissue. Because of this, the living tissue is exposed now. We have to go up to the pericycle level. Now the same thing, we will cover it with little soil, moss, and then tie it with polythene bag. Polythene bags are normally perforated so that air can move in and out. So, this is just covered. Because we have removed the dead tissue, living tissue is exposed here. So, from this living tissue grow adventitious or lateral roots. Now, once the roots are formed, you can cut it from here and this can be used as a new plant. Because everything is done in the air above the ground level, it is called ground layering. So, the advantage of this is that you get a new plant of the same variety, same quality as that of the parent plant. Here also, because it is the vegetative part which is helping in reproduction, they would divide by mitotic divisions. So, all the cells of the new plant would be same as that of the parent plant. So, if we have say a mango tree or a guava tree and we want 100 trees of the same type which produce the same type of fruit, same size of fruit, same flavor and everything. By this method we can tie maybe 10-15 gooties and every branch or every gootee will give us one plant which is exactly identical to the parent plant. Again, this is artificially done. On its own, plant does not do this. So, this also is a method of artificial propagation. So, three methods, one is cutting, other is grafting and third is layering. One more method which comes under artificial propagation, that is micro propagation. Which is also known as tissue culture. In tissue culture, a plant part is selected. That plant part is known as explant. It is the part which you are going to use for getting a culture or many plants developed from that. What should be this plant like? It should be young and it should be capable of division. So, it should have the division power. So, we can select the tip of the root because the root tip has apical meristem. We can select the stem tip which also has apical meristem. We can select a part, a parenchymatous part which has parenchymatous cells which can again divide. But if you select an old part, then as the cells get older, the power of division is lost. So, you want a younger plant and it should have the power of division also. So, that becomes our explant. So, suppose we take the stem tip. Now, that those cells of the stem tip are grown in a culture medium. The culture medium can be agar, and because we are doing it in a scientific manner, agar is the nutritive medium, and in this we will add some phytohormones. The phytohormones which are added in the beginning are two, auxins and cytokinins. When auxin and cytokinins are added in same quantity in the ratio of 1:1, that is auxin to cytokinin is 1:1, this promotes simple cell division. So, this favors cell division. As a result of this, these initial few cells, they will divide and divide and divide, and what we get here is a mass of undifferentiated cells. This is known as callus. What is callus? Callus is a mass of undifferentiated cells, which is known as callus. Why is this happening here? Because the combination of hormones that we have provided in the medium is 1:1 auxin to cytokinin. Now, we transfer these cells into different mediums. The next medium that we are going to select, and we don't have to transfer the whole callus. We can separate this callus into, or instead of this we can take test tubes. Suppose we transfer it into the test tubes. Now the test tube also has the nutritive medium, maybe again agar. These cells, they have to give rise to a plantlet, a baby plant. Now when it gives rise to a plant, it will produce roots, stems, leaf, everything. What is that structure that we want to grow first? Root first or shoot first? We want the root to grow first so that it can anchor itself, plus it can start absorbing the nourishment from the nutritive medium. So because we are providing all the nourishment, we want the newly formed plant start absorbing this material. So now in this we have agar, which is the nutritive material, plus the same two hormones, auxins and cytokinins. These two hormones, they work in a different manner when their combinations are changed. If it is 1:1, that is equal proportion, it simply promotes cell division, no differentiation. If auxins are more than cytokinin, then it promotes root formation. So now we transfer some cells here. Because of this proportion or combination of hormones where auxins are more, it starts to produce root. So the structure which develops first is root. Now we transfer these cells where roots have developed to another medium. It has agar, and now same two hormones, but the proportion is going to be different. It is going to be cytokinin more than auxin. This combination promotes shoot formation. So what we transferred here was few cells with their roots, and now the shoot formation and the leaves, shoots and leaves, a plantlet is formed. We are using the same two hormones. If it is in equal proportion, it promotes only callus formation by cell division. If auxins are more, root formation. Then if cytokinin is more, shoot formation. Now these baby plants or plantlets which are formed, they are all identical to this X plant, because we selected vegetative cells, could be from stem tip, root tip or leaf, and the divisions which are taking place are all mitotic divisions. And we can get hundreds and thousands of plants in a shorter period of time. Plus the space which is required to do this is very less, because you can grow the plant in a test tube. So basically you need test tube stands. So you can have thousands of plants growing in a small area or in a small lab. Now are these plantlets ready to be put in the soil, in ground? Because ultimately you are growing it so that you can grow all these plants in open land. Up till now these plantlets or baby plants were growing in a controlled medium. We were controlling everything because it was in laboratory condition. Temperature is optimum, pH is optimal, nourishment is provided. So there is nothing which is missing in this case. Now suddenly when you shift it into open environment, the temperatures are variable, water content is variable, pH of the soil is variable, nutrients are variable. So can these plants survive in those harsh conditions? Up till now they were growing in a controlled environment. So these plants are given a treatment which is called hardening. Hardening is basically to make them adjust to variable conditions. What is done is, in hardening, 2-3 things. Put them in dark. So these plants are put in dark and then shifted to light. So there is a sudden change, dark and light, dark and light. So into dark and then in light. So this is one thing where they are given alternate. It is basically a shock treatment which is given to these plants so that they get adjusted to the real harshness which they are going to face later on. Plus, there can be a temperature variation which can be given to the plant. It can be kept at little low temperature and then to the warmer condition. So, these two things actually make these plants suitable so that they can survive in the harsh reality of the open environment. Up till now, they were in controlled environment. Before they are put in the open soil, they are given this treatment. So, what are we getting? We are getting multiple plants of the same type as that of the parent plant or the X plant from where you selected that particular cell or cluster of cells, and you can get hundreds and thousands of plants. This method is cheap in the sense you can get many plants in less period of time and less space. But normally this is done for experimental purposes because everything has to be done in a controlled environment. So, this is tissue culture or micro propagation. So, now this is also an artificial method because we are doing it. So, asexual reproduction by the methods of fission, budding, spore formation, etc., plus vegetative propagation. In vegetative propagation we saw natural methods also, artificial methods also. This is one more artificial method. Now, let us quickly talk about sexual reproduction also. We have done everything on asexual reproduction. Now, we will discuss sexual reproduction, then we will talk of parthenogenesis, and then we will also study which part of the life are we discussing. We are just talking about reproduction. So, that we will take up after we are done with parthenogenesis. So, next are sexual reproduction and parthenogenesis.